Hi, it's Mr. Bennett. In this video, what we're going to talk about is we're going to talk about linked genes and we're going to talk about gene maps. So first off, let me give you an example of a linked gene. Linked genes are traits that usually show up together. Let me give you an example in humans. Classic example of a linked genes in humans is blonde hair and blue eyes. This is very, very common. Usually when you see someone who has blonde hair, you expect them to have blue eyes. This is an example of a linked gene. The flip side of this is it's very rare to find someone who has blonde hair and brown eyes. This is something you don't see very often. These are not linked genes. And so the concept of the linked gene is why is it that some traits are usually found together? That's what we're going to be talking about in this video. Now, before I can answer that question for you, we got to go back and we got to review some of the stuff that we talked about already. And one of the first things we got to review so we can understand why this happens is we've got to talk about the process of meiosis again. Now, meiosis, of course, is the process we use to make our gametes, okay? So for guys, we're talking about making sperm, and for girls, we're making eggs, okay? And the process of meiosis is it's basically mitosis twice. It has all the same phases of mitosis, prophase, metaphase, anaphase, and telophase. But whereas in mitosis, we only go through these four phases once, in meiosis, we go through them twice. And the end result of this is if we start out with a cell in humans that has 46 chromosomes, the gametes that are going to be produced are only going to have 23 chromosomes. Of course, the purpose of meiosis is to make gametes that have half as many chromosomes as our normal cells. So that way, when these gametes are used to make a kid, half of the chromosomes come from mommy, half of the chromosomes come from daddy, because the gametes from mommy and daddy each only carry half of the possible chromosomes. Now, to understand about these chromosomes and what's going on, the question comes in, how then do we divide the chromosomes? Because when you make your gametes, you only pass on half of your possible chromosomes, okay? Humans have 46 chromosomes, and when you make your gametes, you're only going to pass on 23 of them. Why do we only pass on half? How does that happen? Well, the first thing you got to understand about human chromosomes is this picture you're looking at right here. These were actual microscope pictures taken of human chromosomes. And the one thing you'll notice right off the bat is they're paired up. Okay? Yes, we have 46 chromosomes, but humans only have 23 different chromosomes. Okay? And for each of these 23 chromosomes, we have a pair of them. Okay? So that's where the numbers come in down here. Okay? All the way through, and then of course, our 23rd different chromosome is going to be the sex chromosomes X and Y. So we have 46 chromosomes because for each possible chromosome, we have two of them. So here's chromosome 1 right here, and you can see we have two of them. We have two copies of chromosome 1, we have two copies of chromosome 2. Now, you may have already been able to guess the reason why we have two copies of each chromosome. The reason why we have two copies of each chromosome is we got a copy of each chromosome from mom and dad. So for chromosome 1, your mommy gave you a chromosome 1 and your daddy gave you a chromosome 1. For chromosome 2, your mommy gave you a chromosome 2 and your daddy gave you a chromosome 2. And so that's why we have 46 chromosomes. We have 23 different chromosomes and we have two copies at each of these. Now, what it comes down to when we're dividing up our chromosomes in order to make our gametes is we're only going to pass on 23 chromosomes, but we're going to pass on one copy of each of the 23 different chromosomes, okay? So when you make your gametes, each one of those gametes is going to have chromosome 1, chromosome 2, chromosome 3, chromosome 4, all the way through chromosome 23, which is your X and Y chromosome. Okay? So every one of your gametes carries one copy of each of the 23 chromosomes. Now, what it comes down to is which one do you pass on? Let's say we're talking about chromosome 9 here. Okay, you got a copy from mommy, you got a copy from daddy. Which one do you pass on? Well, about half of your gametes are going to have a copy of each. 50% of the gametes you make are going to carry the copy of chromosome 9 you got from mommy, and the other 50% of your gametes are going to carry the copy of chromosome 9 you got from your daddy. So hopefully this helps you understand a little bit more about how we divide the chromosomes up. It's completely random, and for each of the chromosomes, we're going to pass on either the one we got from mommy or the one we got from daddy. Okay, so what I just tried to explain to you on the previous slide, we have a term for that in biology, and the term that we use is independent assortment of chromosomes. This is the term that describes how chromosomes are divided up during meiosis. So as mentioned 100 times, every time you make a gamete, that gamete is only going to have half of your possible chromosomes. You have 46, your gametes are going to have 23. Now, you're going to receive, each gamete is going to have one copy of every chromosome you have, 1 through 23. And which chromosome it gets, it's either going to get the one it gets from mommy, the one you got from mommy, or the one you got from daddy. So let's work through this picture here because, there's a lot going on in this picture. So what we got going on here is we got a species that has 3 chromosomes. Here's 1, here's 2, here's 3. Okay? Here's the dad over here. That's what the word paternal means. Here's the mom over here. That's what the word maternal means over there. Now, this individual right here, their offspring, he has one copy of chromosome 1 that he got from his daddy. He has one copy of chromosome 1 that he got from his mommy. He's got one copy of chromosome 2 he got from his daddy, one copy he got from mommy, and so on and so forth that way. Now, of course, how these chromosomes came together is the sperm from daddy combined with the egg from mommy. When the sperm and the egg combined, these chromosomes paired up with these chromosomes here, making our individual. That's what we got going on, right? So we got our individual here. He's got chromosomes from mommy, and he's got chromosomes he's got from daddy. The purple ones, of course, represent the ones he got from daddy. The green ones represent the chromosomes he got from mommy. Now, when this individual makes his gametes, every one of these gametes has to have a copy of chromosome 1, chromosome 2, chromosome 3. 1, 2, 3. 1, 2, 3. And off we go. Now, what we mean by independent is which copy of the chromosome gets passed on, either the one he got from daddy or the one he got from mommy, is completely random. So it is possible for this individual to make a gamete that only has the DNA he got from dad. It is also possible for him to make a gamete that only has the DNA that he got from mom. Okay, it is possible for this guy to make a gamete here that has the copy of chromosome 1 he got from dad, the chromosome 2 he got from dad, and the chromosome 3 he got from dad. Same thing with mom. Or it's possible that he makes a gamete that has chromosome 1 and 2 from dad, but chromosome 3 from mom. Or chromosome 1 and 3 from dad, but chromosome 2 from mom. And you can see from these pictures that even though this species only has 3 chromosomes, there are 8 possible chromosome combinations representing the gametes that this guy can make. So that's what we mean for independent assortment of chromosomes, is you're going to pass on the chromosome you got from mommy or daddy, and that leads to a whole bunch of possible chromosomes. All right, so knowing that chromosomes are passed on independently of each other, which copy of chromosome 1 you pass on has no effect on which copy of chromosome 2 you pass on. This leads us back to Mendel's law of independent assortment. Independent assortment was the idea that what you get for trait A has no effect on what trait B is going to be. Well, the law of independent assortment works because traits are often found on different chromosomes. And so because the chromosomes are passed on independently, that makes it so the genes are passed on independently. So using this simplistic example here, we got a species that's got 2 chromosomes, G and Y, and it's got 2 copies of each of these 2 chromosomes. So you're going to have traits that are going to be on the G chromosome, and you're going to have traits that are going to be on the Y chromosome. Now, which version of the G and the Y chromosome this organism passes on is completely random, and you can see all the possible combinations we got going on here. Well, where this comes home is if we have a gene that's here on the G chromosome and it gets passed on, it's going to be passed on completely independent of genes that are on the Y chromosome, because the G's and the Y's are passed on independent of each other. So you may get this gene here on this G chromosome, but that's going to have no effect on which version of the Y chromosome you're going to receive. Okay, so let's go back to humans, and let me give you an example in humans. All right, so here is our chromosomes for humans. We got the 23 different versions of chromosomes all paired up. Now, the gene that causes blue eyes is found on chromosome 15. On chromosome 15 is where you're going to find the gene that causes blue eyes. Well, the gene that causes brown hair is over here on chromosome 16. So when you make your gametes, we've got this copy of chromosome 15 we got from mommy, we got this copy chromosome 15 we got from daddy. 50% are going to receive of your gametes are going to receive the chromosome from mommy, 50% are going to receive the chromosome you got from daddy. That has no effect whatsoever on which version of chromosome 16 you pass on. So if you pass on your chromosome 15 you got from mommy, that doesn't make you more or less likely to pass on the chromosome 16 you got from mommy. Or it doesn't make you more or less likely to pass on the chromosome 16 you got from daddy. So, because these two traits are on two different chromosomes, whether or not you get the blue-eye gene has no effect whatsoever on if you get brown hair, because they are on different chromosomes and they're going to be passed on completely independent of each other. So if traits are passed on independently of each other, why is it then that blonde hair and blue eyes are often found together? Well, the law of independent assortment applies in 90% of the cases of genetics, but the law of independent assortment only applies for genes that are on different chromosomes. If you have two genes and they're on different chromosomes, they're going to be completely unlinked, and it's going to be completely random which of those two traits you get. But if you're talking about genes for a trait that are on the same chromosome, these traits are considered to be linked, and therefore they are more likely to be found together. And blonde hair and blue eyes is an example. The gene that causes blonde hair is on chromosome 15, and the gene that causes blue eyes is also on chromosome 15. So because these genes are on the same chromosome and we pass on whole copies of our chromosomes, that makes it so these genes are usually likely to stick together. If mommy gives you a copy of chromosome 15 that's got the blonde hair and it's got the blue eye gene on it, well, guess what? When you pass on your chromosomes, you're going to pass on that copy of chromosome 15 you got from your mommy, and it's very likely that that blonde hair, blue eye gene is going to, those blonde hair and blue eye genes are going to stay together on the same chromosome and get passed on to your kids together as a package deal. All right, so this brings us to the concept of linked genes. And in the cases of linked genes, the law of independent assortment does not apply. We throw it out. This is a non-Mendelian case where independent assortment does not apply. Now, this only happens on genes that are found on the same chromosomes. So the idea behind linked genes is genes that are located close to each other on the same chromosome are more likely to be inherited together. It doesn't always happen, but it's far more likely to happen. So if we look at this picture we got going on, let's say this is chromosome 1 over here, and this is chromosome 2 over here. So we got two different chromosomes. Well, gene 1 here is only found on chromosome 1, and it is going to be inherited completely independent of 2, 3, and 4. We're not going to see any linkage between gene 1 and genes 2, 3, and 4 because they're on different chromosomes. But these three genes right here, 2, 3, and 4, we would expect to find them together more frequently because they're all three of them on chromosome number 2. Okay? And some examples of linked genes that you find in humans, we've already talked about blonde hair and blue eyes a lot. Red hair and freckles, the gene that causes red hair and the gene that causes freckles, they're both found on chromosome 15. And so that's why you see lots of redheads with freckles. Other thing you see going on is in plants. Flower color and the shape of the pollen. These are linked genes, and the gene that causes this trait is both found on the same chromosome. So that's a concept of linked genes or linked traits. Traits that are more likely to be found together are usually found on the same chromosome, just like blonde hair and blue eyes are found on the same chromosome, chromosome 15. All right, so let's talk a little bit more about linked genes. So linked genes is the idea that traits that are caused by genes that are found on the same chromosome are more likely to be inherited together. Well, how close together they are on the same chromosome actually has an effect on how closely linked these traits are going to be. That these two genes right here are going to be more closely linked than these two genes over here because they're closer together on the same chromosome. On the flip side, if you have a gene here and you have a gene here, even though they're on the same chromosome, you may not find the trait caused by these genes to be very linked because they're so far apart from each other on the same chromosome. And the reason for this is a process called crossing over, which we're going to cover next. All right, so what the freak is crossing over? Crossing over is something that happens during meiosis, and this is an actual picture here of a chromosome going through the process of crossing over. And what happens during crossing over is the chromosomes exchange pieces. They exchange pieces of each other. And so what you got going on is the DNA you receive from mom and the DNA you receive from dad gets all mixed together. All right, so let's say this is chromosome 1 here, and here we got the copy we got from mom, and here we got the copy we got from dad, okay? This is what mom and dad gave you, but you're not going to pass that on. When you go through the process of meiosis, you actually mix up the DNA you got from mom with the DNA you got from dad. So here you got this picture here. You can see why we call it crossing over, because the chromosomes cross over each other. And the result of this, when crossing over is done, this piece of the chromosome right here used to be nothing but DNA from mom. But after crossing over is done, about 60% of this piece of chromosome here came from mom, and 40% of this chromosome came from dad. We shuffled the DNA together. This one here, you can see here we got a piece of chromosome we got from dad. This little piece of chromosome right here, that when we got it from our parents, it was 100% DNA we got from dad. But when we pass it on to our kids, you see what's going on here is this chromosome that used to be all from dad has got this piece right here that came from mommy. Here's how I think about it. You can think of the DNA that you got from mommy as being a red deck of cards, and you can think of the DNA you got from daddy as being a blue deck of cards, all right? That's the DNA that you got. Before you pass it on to your kids, you take that deck of cards from mom and that deck of cards from dad and you shuffle it up, and then you pass it on. And so your kids are going to get a mixture of blue and red cards, just like when you pass your chromosomes on. Your chromosomes are not the way you got them from your parents. You shuffled them up. And so now this copy of chromosome 1 that you got from mommy has now got pieces of DNA from mommy and from daddy. So, an example of crossing over at work is a chromosome could have an eye color gene from mommy, let's say the gene that causes eye color up here, and that's going to be from mommy, and a gene that causes hair color is down here, that came from daddy because you shuffled it all up. Now, how does this play in with linked genes? Well, if you look at these pictures here, you can see the whole sections of the chromosome get passed on. And so the closer the genes are to each other, the more likely they are to move as an entire section when they get mixed up. And that's the reason why a gene that's up here and a gene that's down here are very unlikely to be linked to each other. They're more likely to get mixed up because you can see no example in this picture where this section of the DNA and this section of the DNA moved together to form a new chromosome. They got mixed up. So if you look at this one here, this gene up here is still there, but down here, now we're talking about the gene we got from daddy. They got separated because these two genes are so far from each other. So that's crossing over. That's the mixing of the chromosomes together. It makes it so genes that are closer together on the same chromosome are more likely to be linked. Genes that are further apart from each other on the same chromosome are less likely to be linked. All right, so how do we know this? This is where we come to the concept of gene maps. The scientists have been busy at work trying to figure out each and every gene that causes a trait, and then where on what chromosome that gene is found, and where on that chromosome that gene is found. So, gene maps. Gene maps are a tool we use for studying genetics, and what gene maps show you is which chromosome a gene is found on, and where on that chromosome that gene is found. I told you the blonde hair and blue eyes are found on chromosome 15, and there's a gene map for that if you looked at the gene map, each of these things here represents a trait. And so you could find a gene map. This gene map doesn't have all the possible genes for a human. It just has a few of them. If you looked at a gene map for chromosome 15, you could find where the gene is. We've located where that gene is. It's on chromosome 15, and exactly where on chromosome 15 that is. Now, the reason why we care about that is, let's come over here and let's look at chromosome 2. Here's two genes right here that I'm not going to bother to read the name of. We would expect these two genes here to be linked because they're close together and they're on the same chromosome. We would not expect these two genes to be very closely linked to this gene here on the opposite end of chromosome 2. Because they're so far apart, they're more likely to be separated during the process of crossing over. And of course, these two genes here are going to be completely unlinked from this gene here on chromosome 3 because they are on different chromosomes. So that's the process of linked genes. This is the reason why blonde hair and blue eyes are usually found together, because they're both on chromosome 15. They're found very close together, so they're not likely to be separated by crossing over on the same chromosome. So when you pass them on, you're usually going to pass on both of these genes at the same time. Whereas genes that are on different chromosomes, that's where the law of independent assortment applies. And then, of course, we just talked about gene maps. Gene maps is where we can look and we know which gene, for every gene, we know where it is, on which chromosome, and on what chromosome, and where on that chromosome to find it.